All right. Welcome back, everybody. Thanks for joining us for another session of the Australasian Plural Seminars, our monthly series where we talk about all things plural from faculty, local and national, international. So thank you for everybody joining us. Today, we're honored to have Dr. Ken Chan join us from the Chinese University of Hong Kong to discuss advances in molecular diagnostics for tuberculosis pleuritis with his recent publication in the New England Journal. So we're really excited to have him chat about that and give us his thoughts on TB pleuritis. So without further ado, I'll turn over the mic to him and get him started.
Yeah. Right. Hello, everyone. Thank you for having me here, and very thankful for the kind introduction by Chairman. And all right, can you see my slides? Yeah, should be okay. Now, sorry for a bit cracky voice, just recover from URTI. Now, today I would like to share about the advances in molecular diagnostic for tuberculous pleuritis. In Hong Kong, we do have an intermediate burden of TB, so we need something that's accurate to include or exclude this diagnosis. So here are my disclosures, nothing related to the current talk. Now, I will just skip all those basic things about TB pleuritis, or in short TBP. If you want to know more, I advise you to go back to the tab series about it by Jane Shaw, available on YouTube. So a very brief summary about TB pleuritis. This is one of the most common types of extrapulmonary TB, and it's actually a type 4 hypersensitivity reaction of the pleura towards the microbacterial antigens. Therefore, you can't find a lot of MTB bacilli in the pleural space, which is known as a polybacterial nature, which makes the diagnosis of TBP is very challenging. Therefore, most of the time, the diagnosis is by a composite of microbiological, histological, and clinical features. Now, there are many challenges to diagnose TB. First, it has heterogeneous presentation. Some TBP cases may present without systemic symptoms, without apparent lung parenchymal infiltrates. And the effusion itself can be neutrophilic in some cases, not the classical lymphocytic nature. And the sensitivity for peripheral AFP smear is low. And even you do a TB culture, the long turnaround time will just delay the diagnosis. Biopsy may not be opted by some attending physician because of invasive nature and lack of expertise or lack of support. And to me or to some physicians, we do prefer a direct confirmation of MTB bacilli. ATA itself is good, but it's not a direct confirmation, and it cannot inform us about the drug resistance. And therefore, with all these challenges, the diagnosis can be delayed and therefore delayed in the initiation of the treatment. So this is a very nice review by Jane that was published a few years ago on the proofread MTB culture. You can see that even with liquid culture or the pleural tissue culture, the sensitivity is not very optimum. At most, it's around 50 to 70% for the MTB culture, depending on the specimen we are using. So can we just improve the diagnostic pathway, making it less invasive, avoiding pleural biopsy? Can we confirm a direct confirmation of the TB bacilli in the pleural space? If a test is highly sensitive, specific, and make good use of the remaining pleural fluid. And we now have advanced technology of molecular tests. Can we combine all this together to fulfill all these targets? So before introducing the latest evolution of the molecular technology for TBP diagnosis, I would like to introduce you three important gene targets for MTB. So these are the three gene targets: IS6110, IS1081, and DEPR. These are MTB genome unique with variable copy number in the genome. The full length is more than 1,000 base pair, but the typical length requiring for PCR amplification is around 100 to 200 odds base pair. So I have selected some key papers about the about the molecular diagnosis. This is one of the biggest cohort, multi-center cohort done in China. They enrolled patients prospectively and sent the proofread for Expert Ultra. You can see that this is a summary. They centrifuge the proofread, get the sediment, apply the Expert Ultra to the sediment, and actually the Expert Ultra, they read the long sequence IS16110, IS1081 in the sediment. The sensitivity is only around 44% with very high specificity. Now, the sensitivity is actually a bit different for those definite or probable TBP. If that's a proofread MTB culture positive, the sensitivity is higher. But whenever there is negative TB culture in the proofread, sensitivity drops significantly. With the high specificity, if there is a positive test, you can say that's positive TBP. But negative case, because of the low sensitivity, a negative result could not tell you whether it's a truly negative or false negative. So the problem of low sensitivity is believed that the amount of genomic DNA of TB is of low amount in the sediment. And this is also recapped by Jane in the review that the sensitivity is around 50% for TBP when using Expert or Expert Ultra. So subsequently, with further advancement of understanding, we know that we can check the cell-free DNA in the supernatant. Here are the 3 key papers that was published a few years ago, from China and also from India. Now, during the centrifugation, we actually separate the fluid into sediment and supernatant. Further understanding of the disease know that the amount of cell-free DNA is abundant in the supernatants. When we apply the PCR and target it along the IS6110 sequence, we can actually achieve a higher sensitivity at around 79%. For those cases with culture positive TB Pleuritis, the sensitivity can be very high, up to 96%. But again, the sensitivity drops in those culture negative cases. So we're actually doing better by targeting a different material or targeting the supernatant. So can we just boost up the sensitivity? This is an important paper from Thorax, published last year, also from China. They optimized the molecular techniques with 2 important things. The first is by ddPCR, droplet digital PCR. It's a physical partitioning. Now, it was believed that there was much inhibitory action on detecting the TB DNA in the fluid because there are a large amount of human DNA surrounding the TB DNA. So with the ddPCR, we separate each droplet fluid into 20,000 microdroplets with individual PCR performed in each microdroplets. And therefore, this can reduce the interference from the human DNA and potentially improve the pickup rate for the TB DNA. The second thing is that they use a shorter amplicon length. Further understanding of the TBP found out that the length of the cell-free DNA fragment and the proof rate is actually around 60 to 80 base pair. While when the older generation of the PCR always targeting a longer length of base pair, more than 100, a large chunk of the TB cell-free DNA actually missed. Therefore, in this study, they use a shorter amplicon length to target DNA fragments with a 60 or 80 base pair amplicon length. And actually, they can do a better performance with pooled sensitivity around 85%. Again, you can see a very high sensitivity in those definite TBP cases, with a slight drop in the sensitivity for those probable or culture negative cases. So we now understand that the cell-free DNA is more abundant in the superlatents. We need to target the short gene fragments. And we are now having next generation sequencing instead of the quantitative PCR. So can we combine all these together and improve the sensitivity? However, still there one problem we still need to overcome is the high sequence similarity between TB and non-tuberculous mycobacteria. Whenever we're doing next generation sequencing, we're amplifying all the DNA fragments in the specimen of variable length. Potentially, we may also amplify those DNA fragments from contamination from surroundings or including those NTM contamination. So we need to do something to overcome this problem. So with the funding from government, from NGO, and also collaboration with other teams, including microbiology and chemical pathology, we conduct the study, Midnight Study. That is a diagnostic utility of Mycobacterium tuberculosis cell-free DNA in TB and non-TB pre-fusion. I put a firefly there because I always think that finding TB bacilli in the periphery is just like finding a firefly with a very dim light in the midnight. You know there is the firefly is somewhere there, but you can't really localize it. So we have two theological advancement in this study. The first thing is the cell-free DNA fragmentomics. Now, whenever the DNA is degraded or fragmented in the fluid, the DNA is cleaved by a human enzyme. So the DNA fragment will have different nature or different characteristics, including the size, the morphology, and also the ACTG ending. The different type of ACTG ending, what we call end motif, actually carries some meaning. This is a paper of analyzing the fragment ends of circulating microbial DNA in patients admitted for sepsis. We found that those DNA fragments ending with GG and CC ending or CCN motif are associated with general infection. Those with other endings are associated with contamination. So with this type of pattern and motif pattern, we can kind of separate those patients with true TB infection and from those NTM contamination. The second advancement is the bioinformatic alignment algorithm. We compare the NTB genome with more than 2,500 reference genomes, including those with NTM. So we compare the genome. Those genomes that is matching between these two genomes or these two group of genome will be considered as non-TB specific. Those not matching and only present in the MTB genome is considered as MTB specific. So we use those MTB specific genome as a target and what we call a smart MTB genome. Instead of those IS6110 gene targets, we use the whole genome specific as the reference genome for the test analysis. So we take the pleural fluid and amplify them with NGS, and then we'll align them with the mass MTB genome. Those matching with MTB genome will be considered as MTB-derived DNA. So with these two technical advancements, we published the paper in NEJM Evidence in March. We prospectively enrolled, screened more than 1,200 patients, and we exclude those patients with history of TBP, reason is poor instrumentation, and only include those patients with new onset electrical refusion with known diagnosis. And finally, 329 patients included cohort, and around 1/10, 34 patients had TBP confirmed. So we separate these patients into different groups, including TB, fluid overload, primary diffusion, malignant diffusion, etc. We apply all the peripheral sample for targeted MTB sequencing with our technology, MTB culture, PCR, and ADA. And here are the results. With the targeted sequencing of the periphery, we found that the level of MTB cell-free DNA fragment is actually much higher in TBP group and at a very low level in non-TBP group. The AURC is 0.9996, approaching 1. So this is the diagnostic performance and the diagnostic cutoff at 2 reads per 10 million. The sensitivity of the target sequencing was 97%. In the same cohort, the culture, the sensitivity for culture, PCR, and ADA were listed here at 47, 26, and 61% respectively. So you may recall that in those cases, in previous cohort, those cases with culture MTB, the sensitivity of those tests will be lower. In our cohort, the sensitivity remains at a very high level. While you can see that the sensitivity will drop for PCR and ADA. So based on these studies, we have a very high sensitivity of picking up TBP. And actually, this technique can have some additional utility that we can further explore. The first is that the cell-free DNA can be detected before the diagnosis. We have two cases with recurrent cardiogenic perfusion. This one patient, the first patient had tapping done, and we checked and found the cell-free DNA level at 51, while at that episode was still transudative. But then 8 months afterward, the patient had a positive MTB culture and PCR during the second recurrence of the cardiogenic perfusion. So we consider this case as true positive. And patient two had in index MTB cell-free DNA level at 4.34 reads per 10 million. And then he had recurrent cardiogenic effusion afterward, or transudative. At the time of publication, we consider this case as a false positive, but after publication, the patient later finally agreed to have proof biopsy and confirmed TBP. So this case later we should be a true positive. And the second utility is potentially it can detect resistance-associated variants. We include 16 TBP samples with the MTB DNA reads that cover at least one of the gene targets or gene regions containing the drug resistance as advised by the WHO. And we could not find out any resistance-associated mutation. However, this findings is limited by limited number of sample, and we don't have any drug resistance case at the moment in the cohort. And third utility is that we apply the same technology to the plasma. Out of the 28 plasma available, out of the 31 plasma available, we can find MTB cell-free DNA in plasma in 28 samples, which is also much higher than those with in the non-TBP cases. The AUC was approaching to 0.95. So this is from summary. Actually, you can see the progressive improvement of sensitivity with different generation of the molecular techniques. And now with the latest technique, we can boost up the sensitivity to more than 95%. So with the midnight study, the midnight is actually now illuminated. So in the past 15 minutes, I've walked through the heterogeneous presentation of TBP and the diagnostic challenges. And with the leap of molecular diagnostic technology, we can catch all the firefly and no longer lost in the dark. So thank you. I would like to also thank for my clinical research team and the support, and welcome for any questions.
Brilliant, Ken. Thank you so much for that review. Amazing to see the evolution of the technology and kind of where we stand now with your latest work. So with that, I'll open up the chat for questions. Please feel free to just type anything in there and we'll start reviewing those and asking Dr. Chan about his thoughts and about whatever may be kind of on your mind after this the talk. Ken, just to kind of start it off from my perspective is, so what kind of led you down this path? I mean, obviously, besides the clinical need, for better studies, what was it that kind of drove you to do this work, and how did you get started?
The primary intention is to minimize the need for probe biopsy. Because our center, our team has a lot of problem when dealing with those patients with perfusion. Because in Hong Kong, some patient may present present to the medical department instead of respiratory team, so they would just do the tapping and without doing any probe biopsy, and then they would just drink all the fluid dry after the first tablet. So that would create a lot of difficulties when we want to do further probe sampling. And that's why we want to have some advanced technology of liquid biopsy. And in our center, the Chinese University of Hong Kong is well known for the technology of liquid biopsy. The chemical pathology team invented this technique and applied to cancer screening and also non-invasive prenatal screening. So we try to combine the clinical need and the expertise, and we need to choose one disease that will have clinical challenges. And that's why we choose TBP as the potential target. And fortunately, we did have some success in the technical advancement. And that's why we include we start a cohort and get some funding and proceed just proceed further.
No, that's amazing. And you're exactly right. I mean, it's so challenging in terms of even pleural biopsies, even if it's ultrasound guided or anything like that. And frequently we see that patients will have undergone thoracentesis, fluids all drained, and now we have nothing else left, right, to do about it. So even if we wanted to biopsy nodule, it just makes it more challenging and risky.
Yeah.
And the morbidity of doing a thoracoscopy obviously exists in those situations where you don't have any more fluids. So I mean, it's great that we can use all these techniques, and I think it's again, kind of local expertise, building upon what you have and applying it to a solution. So it's great that you've been able to, really look at this and develop it with the local expertise that you have, and I think that's really a lot of times what it takes. So for any of our younger members and that are joining us today, it's really just a lot of it is just understanding what's available. The experts in your center, because you're not going to have experts in every single thing, right? But there's definitely going to be people that are be exceeding and doing really well, and really just kind of collaborating with those people to then come up with a solution to something that's actually a huge need, So kudos for you for doing that, identifying that, and working with your team. This is going to be really game changing for all our TB patients. So. In terms of your liquid biopsies, kind of when do you anticipate that it'll be readily available and kind of accessible to people that we're actually going to need outside your university setting?
Well, the chemical pathology team has grant patent to the technology. I think they will try of marketing it after some validation. And actually, we have applied two different government funding on validating this test. First is prospective validation, whether we can reduce the need for probe biopsy clinically. And second, whether this test can be applied to other areas with different TB burden. So we need to make sure that the test itself is robust enough in those areas with low or very high TB burden. Okay, make sure that we are not doing something extra or excessive, right?
Absolutely. No, great. And questions coming through the chat. So, did you observe any correlation in DNA levels with other surrogate markers such as ADA in your study? And you kind of answered this, but what do you think should be the next step ahead, RCTs or such?
Yeah, thank you for the question from Jean. Yeah, we try to correlate the DNA level, but we could not find any meaningful association between the DNA level and the patient characteristic or the disease characteristics, including ADA level. And also including the culture status is all not affecting the DNA level as well. So in next step, as mentioned, we just need to validate it. We don't have any roadmap at the moment to put it in the market. I think we just dependent on the chemical pathology team because they have the patent there.
Hmm.
But if it will be available, it would be a very we need to position it at a right pace, whether we do it upfront or as a second line test. Because sequencing itself is very expensive. At least in Hong Kong, I'm not sure the price or cost in Australia or other places, but in Hong Kong it's very, very expensive. And the turnaround time, even at a fast, I mean, in a expert center, it may take a few days or even a week or so. So I would say whenever doing this, we need to take the cost and the local workflow into consideration. Like, for example, if in our center, we need to educate the colleagues not to drink all the fluid out first. Do a tapping. If it is very apparent that from the first tapping you get the diagnosis, you may not need this liquid biopsy. But if the patient, you don't have the diagnosis from the first tapping and you are planning for second tapping plus biopsy, I will advise to get the fluid for the liquid biopsy as well. First, biopsy is not always accurate for TB, and it may still has a false negative rate, although usually lower. And the liquid biopsy itself can improve the diagnosis and improve the sensitivity. I hope in the future we can optimize this technique not only targeting the TB, but also use the same technology for other diagnoses, including malignancy. And actually, the chemical pathology team has some other validated tests for liquid biopsy in diagnosing malignant pleural effusion. So I would say all these well, cost demanding tests or advanced tests can be put at well as second line. But it would require some prospective study for validation, confirming its utility.
Yeah, no, absolutely. And you're exactly right in terms of, these costs of all these studies. And it's great to have all these things, but you're exactly right in a center where you don't have just a ton of money to throw around, where you have to really be cost conscious and utilize everything else beforehand. And so, yeah, it makes sense that you would use this as a second line, if your initial preliminary testing, when you have a pre-test, high pre-test probability, it doesn't really yield an answer for you, then utilizing this, again, makes sense that you would do it before you do something more invasive, whether it's a biopsy or thoracoscopy of some sort. So that's really, it's great to have just yet another tool in the arsenal for helping manage these pleural disease. And it's interesting to show, the second case that you had, it's great to see that, the test was valid even in that patient that initially you thought was a false negative. When you confirm with the biopsy that you truly did have, some utility to the test. So it's great to be able to showcase these kinds of things. But again, like you said, larger studies, validating it, prospective studies ideally. And then the other important thing I think you said was, really in low incidence areas is it all still hold up? Because that's going to be the other areas where people may just not be comfortable, coming from the States. Although we had a TB center where I trained, kind of around the country, it's not something that we see all the time. And so I think for places where you actually have low incidence, it may hold a little bit more value there anyways, because people just aren't necessarily used to the presentation, aren't used to the clinical kind of components that, you would analyze there, right? Where, you go to anywhere Southeast Asia where there's a high prevalence and you're like, oh, absolutely, this is TB. It's just not part of the differential in those areas. And so I think, for places like the US or other places where there's just a lower prevalence, I think that would be another place where it'd be very beneficial to have a liquid biopsy like you've developed.
Yep, agree with that.
Yeah. Okay. Very good. Well, I think we're right on time then. So if there's no more questions, thank you so much, Ken. I really appreciate your
Thank
time.
you.
Congratulations on your great work, and I look forward to having this available for our patients. And for those that may have joined late or joining now, please do remember that these are recorded sessions. We have them on YouTube, so please join in and you can see what you've missed and always send an email or contact us if there's any other questions that are burning. But please do join us next month where we have Dr. David Feller-Kaufman joining us here and look forward to seeing everybody then. Thanks again, everybody.
Thank you. Bye.